friends to a new session on genomics and proteomics. In this session, we will discuss cytogenetics, chromosome painting, fluorescent in-situ hybridization or FISH, micro-dissection and flow sorting. Cytogenetics is the study of chromosomes and their role in heredity. It deals with chromosome structure and composition, the methods used to analyze chromosomes, chromosome abnormalities associated with diseases, the role that chromosomes play in sex determination, and changes in chromosomes during evolution. It includes routine analysis of G-banded chromosomes, other cytogenetic banding techniques, as well as molecular cytogenetics such as fluorescent in-situ hybridization or FISH and comparative genomic hybridization CGH. Over the past decades, versatile methods based on FISH have transformed cytogenetics into a molecular science and provided cytogeneticists with powerful new tools. Conventional cytogenetic testing or routine chromosome analysis, sometimes referred to as karyotyping, is provided for a variety of clinical applications. In FISH procedures, labeled DNA or RNA probes are hybridized with their complementary target DNA sequences on chromosomes. Chromosome painting is a term used to describe direct visualization using in-situ hybridization of specific chromosomes in metaphase spreads and in interphase nuclei. Chromosome painting, competitive hybridization using entire chromosome, specific libraries for chromosomes as probes, and human genomic DNA as the competitor was one of the first applications of FISH. Chromosome painting coupled with FISH is now used routinely to enhance the identification of chromosomal rearrangements, the assignment of breakpoints, and the determination of the origin of extra chromosomal material. These technologies have enabled the development of reverse chromosome painting, in which the paint is produced from sorted aberrant chromosomes and hybridized back onto normal metaphase spreads to identify directly the composition of the aberrant chromosome. Cytogenetic analysis by chromosome painting. Chromosome painting involves the use of fluorescent tagged chromosome specific DNA sequences to visualize specific chromosomes or chromosome segments by in-situ DNA hybridization and fluorescence microscopy. Chromosome painting refers to the hybridization of fluorescently labeled chromosome-specific composite probes to cytological preparations and allows the visualization of individual chromosomes in metaphase or interphase stages and the identification of both numerical and structural chromosomal aberrations with high sensitivity and specificity. The simultaneous hybridization of multiple chromosome painting probes, each tagged with a specific light-emitting fluorochrome, has resulted in the differential color display of human and mouse chromosomes, which is also called color karyotyping. The whole chromosome painting probes are complex DNA probes derived from a single type of chromosome that has been usually flow sorted or micro-dissected, amplified and labeled by degenerate oligonucleotide polymerase chain reaction to generate a paint which highlights the entire chromosome homogeneously along its side. Through this method, chromosome arm specific paints and region specific paints can also be generated. This type of probe is most useful for clarifying cytogenetically visible structural or numerical chromosome rearrangements in metaphase, but painting probes are not helpful in the analysis of interphase cells because the signal domains are so large and diffuse. Fluorescent in-situ hybridization has been used to dictate the location of specific genomic targets using probes labeled with specific fluorochromes. So, the chromosome painting is also called M-FISH or multicolor FISH. The technique allowed the detection of simple and complex chromosomal rearrangements and abnormalities by the conventional cytogenetic banding techniques. Chromosome painting was developed by research teams at Lawrence Livermore National Laboratories and Yale University. Both groups had taken advantage of the availability of cloned DNA libraries that were derived from flow-sorted human chromosomes. Chromosome painting has improved the efficiency of screening cells for chromosome abnormalities, in testing chemicals for mutagenicity and for rearrangements associated with tumors. Painting probes detect chromosome rearrangements. Chromosome painting probes are now available for an ever-increasing number of species, most notably for the mouse and the rat, allowing the expansion of chromosome painting analysis to animal models for human diseases. Fish techniques have been developed and applied to identify the origin of the markers and other structural chromosomal aberrations. The use of chromosome painting probes in 1, 2 or 3 color fish experiments has significantly improved the diagnosis of chromosomal aberrations. Chromosome painting can be used to identify homologous chromosome segments in different species and to map probes of different complexities and chromosome rearrangements in a single experiment. The complete karyotypes of various mammals, including primates, carnivores and artiodactyls, have been analyzed by chromosome painting in recent years. Chromosome painting probes available in liquid format are directly labeled in either a red or green fluorophore, and they can be mixed together to label a number of chromosomes in a single reaction. Fish, principles and methodology. Fluorescent in-Situ hybridization or FISH is a technique in which single-stranded nucleic acids, usually DNA, sometimes RNA, may also be used. They are permitted to interact so that hybrids are formed by molecules with sufficiently similar complementary sequences. Through nucleic acid hybridization, the degree of sequence identity can be determined and specific sequences can be detected and located on a given chromosome. The method comprises of 3 basic steps: fixation of a specimen on a microscope slide, hybridization of label probe to homologous fragments of genomic DNA, and enzymatic detection of the tagged target hybrids. Probes can vary in length from a few base pairs for synthetic oligonucleotides to larger than 1 mega base pair. Probes of different types can be used to detect distinct DNA types. PCR amplified repeated DNA sequences, oligonucleotides specific for repeat elements and cloned repeat elements can be used to detect clusters of repetitive DNA in heterochromatin blocks or centromeric regions of individual chromosomes. Chromosome painting with fluorescence in-situ hybridization or FISH. Cytogenetics entered the molecular era with the introduction of in-situ hybridization, the procedure to locate the positions of specific DNA sequences on chromosomes. Since the first in-situ hybridization experiments in 1969, many variations of the procedure have been developed and its sensitivity has increased enormously. Fluorescence, a phenomenon whereby a chemical excited at one light wavelength emits light at a different and usually longer wavelength, is used throughout the life sciences to study a wide variety of structures and intracellular activities. Today, most in-situ hybridization procedures use fluorescent probes to detect DNA sequences, and the process is commonly referred to as FISH or fluorescence in-situ hybridization. The success of FISH and all other methods of in-situ hybridization depend on the remarkable stability of the DNA double helix. In molecular hybridization, a labeled DNA or RNA sequence is used as a probe to identify or quantify the naturally occurring counterpart of the sequence in a biological sample. In 1960s, researchers Joseph Gall and Mary Love Pardue realized that the molecular hybridization could be used to identify the position of DNA sequences in situ, that is, in their natural positions within a chromosome. Preparation and hybridization process RNA. Cells or frozen tissue sections are fixed and permeabilized to allow target accessibility. FISH has also been successfully done on unfixed cells. A target-specific probe composed of 20 oligonucleotide pairs hybridizes to the target RNAs. Separate but compatible signal amplification systems enable the multiplex assay, up to 2 targets per assay. The signal amplification is achieved via a series of sequential hybridization steps. At the end of the assay, the tissue samples are visualized under a fluorescence microscope. For example, RNA fluorescence in-situ hybridization for Cre mRNA shows genetically identical cells in which expression of the CRE or Cre is epigenetically regulated. KAP104 is a control gene whose expression is not epigenetically regulated. Preparation and hybridization process DNA. First, a probe is constructed and tagged directly with the fluorophores with targets for antibodies or with biotin by nick translation or PCR using tagged nucleotides. Then, an interphase or metaphase chromosome is firmly attached to a substrate, usually a glass. Repetitive DNA sequences must be blocked by adding short fragments of DNA to the sample. The probe is then applied to the chromosome DNA and incubated for approximately 12 hours while hybridizing. Several wash steps remove all unhybridized or partially hybridized probes. The results are then visualized and quantified using a microscope that is capable of exciting the dye and recording images. FISH testing is a method by which an assessment is made for the presence, absence, relative positioning or the copy number of specific DNA segments by fluorescence microscopy. Depending upon the application, FISH can be applied to metaphase chromosome preparations or interphase cell nuclei. Metaphase FISH studies or microdeletion FISH. Metaphase FISH studies are designed to detect changes, that means the microdeletions or microduplications associated with specific phenotypic findings and require specific and focused clinical questions to be addressed via appropriate DNA probe selection. Genetic changes that are detected in metaphase chromosome preparations by this type of testing are limited to position and copy number changes, primarily losses or deletions, and in some instances gains, that means the duplications of the specific chromosomal regions for which the employed DNA probes are localized. Repositioning of these DNA probes from their normal sites must be determined in metaphase cell preparations, that is translocations, insertions, etc. However, these types of study do not rule out other forms of genetic abnormalities, which may include low level of tissue specific mosaicism and or other forms of molecular alterations like single base pair mutations, uniparental disomies, etc. Interphase FISH studies. Enumeration or rearrangements involving specific DNA probes are the only information available from interphase FISH studies. Additional inferences of the chromosome constitution of cells utilized are not possible from interphase FISH studies. Aneuploidy screening in interphase FISH studies does not address structural content of the chromosome detected. The use of prenatal interphase FISH studies as an in vitro diagnostic test has been cleared by the FDA only as an adjunct test to standard cytogenetic analysis. Dual color or fusion FISH studies. This form of testing utilizes DNA probe systems that are designed to detect well-known neoplastic rearrangements affecting two specific loci, but may not detect variant, complex or typical rearrangements involving these loci. Additional secondary clonal rearrangements may not be detected in interphase nuclei. Further characterization of abnormal cell populations by metaphase FISH or conventional cytogenetic methods is highly recommended. Dual color or break apart FISH studies. These studies utilize DNA probe systems that are designed to detect the involvement of specific loci known to participate in rearrangements involving a variety of translocation partners. These types of DNA probe systems can only identify rearrangements involving a single locus and cannot identify other loci that may be involved in the rearrangement by interface analysis. Further characterization of abnormal cell populations by metaphase FISH or conventional cytogenetic methods is recommended. Flow sorting and microdissection. The origin of marker chromosomes that were unidentifiable by standard banding techniques could be verified by reverse chromosome painting. This technique includes microdissection and flow sorting. Chromosome-specific sequences are highly desired in studies focusing on comparative genomics and genomic organization. Specific chromosomes can be isolated from the rest of the genomes by flow sorting or fluorescence activated cell sorting FAC and chromosome microdissection. In flow sorting, chromosomes are sorted using a laser system that distinguishes chromosomes depending on their size and fluorochrome affinity, that means the AT, GC base content, while in chromosome microdissection, chromosomes or chromosomal segments are literally scrapped and collected. In contrast to flow sorting, microdissection allows the small chromosome fragments. It is generally assumed that flow sorting generates paints of greater complexity and coverage because this method allows the collection of around 300 to 500 chromosomes. Chromosome microdissection was originally developed in studies involving Drosophila polytene chromosomes as a way to obtain DNA markers from specific chromosomal regions and was quickly applied to mammalian genomes during the 80s. Only minute quantities of chromosomal DNA are available after microdissection. The quantity of this DNA depends on chromosome fixation and staining. The subsequent development of in-situ hybridization procedures and establishment of degenerate oligonucleotide primed polymerase chain reaction or DOP-PCR allowed the application of chromosomal probes obtained by microdissection in fish. It is very rapid, is less difficult to handle and involves direct PCR amplification of DNA contained in micro-dissected fragments with a universal primer composed of a mixture of oligonucleotide sequences that lacks complementarity to the target template sequences. DNA isolated by flow sorting or microdissection can be used for genomic research, including the generation of chromosome-specific libraries or markers, physical mapping, and cloning of chromosome-specific expressed sequences or genes. Let us summarize the following discussions on cytogenetics, chromosome painting, fluorescent in-situ hybridization, microdissection, and flow sorting. Cytogenetics is the study of chromosomes and their role in heredity. It deals with chromosome structure and composition, the methods used to analyze chromosomes, chromosome abnormalities associated with disease, the roles that chromosomes play in sex determination and changes in chromosomes during evolution, conventional cytogenetic testing or routine chromosome analysis, sometimes referred to as karyotyping, is provided for a variety of clinical applications. In fish procedures, labeled DNA or RNA probes are hybridized with their complementary target DNA sequences on chromosomes. Chromosome painting is a term used to describe the direct visualization using in-situ hybridization of specific chromosomes in metaphase spreads and in interphase nuclei. Chromosome painting, competitive hybridization using entire chromosome-specific libraries for chromosomes as probes and human genomic DNA as the competitor, was one of the first applications of fish. Chromosome painting coupled with fish is now used routinely to enhance the identification of chromosomal rearrangements, the assignment of breakpoints, and the determination of the origin of extrachromosomal material. These technologies have enabled the development of reverse chromosome painting, in which the paint is produced from sorted aberrant chromosomes and hybridized back onto normal metaphase spreads to identify directly the composition of the aberrant chromosome. Before we attend the next unit, please try to answer the following questions. Write an overview on cytogenetics. Discuss chromosome painting. Write short note on preparation and hybridization process of DNA. Explain the principles and methodology of FISH. Discuss micro-dissection and flow sorting. Now the references are Introduction to Bioinformatics, Atwood and Parry Smith, Pearson Education. Bioinformatics, A Beginner's Guide by Jean-Michel Clavier, John Wiley and Sons. Structural Bioinformatics by Philip A. Bourne and Helge Wiesing Wheely. Bioinformatics, Sequence and Genome Analysis, Mount D.W., Cold Spring Harbor Laboratory Press, New York. Hope this session was beneficial for you. See you in the next session. Till then, bye.